Hi, everyone. Thank you for joining us to our second part of our webinar mini-series today, Single-Cell Sequencing for Every Lab. In this part, we focus on applications of single-cell sequencing beyond RNA profiling. The presentation covers targeted single-cell sequencing techniques, adding a time dimension to your single-cell sequencing research, importance of single-cell sequencing in yeast research. This interactive webinar follows by a Q&A section, so feel free to ask your questions by writing into the Q&A section. You can contact us directly for any project-based consultation afterwards as well. The webinar is being recorded and will be uploaded to our YouTube channel in the following days. We have Dr. Wei Long as our speaker today. Wei has earned her master's degree in biotechnology at the Technical University of Braunschweig, Germany, and her PhD in biotechnology from Aachen University, Germany. In 2021, she joined Singleron Biotechnologies as a product manager, where she supports the development of new technologies and takes care of Singleron's product, including instruments and kits portfolio. Good afternoon, Wei. Looking forward to your presentation.
Thank you very much for your introduction. So also from my side, welcome everybody to my presentation. I will start my presentation with a very short introduction to single-cell sequencing. So we usually compare, when we try to visualize single-cell sequencing, we usually compare the RNA sequencing, the normal bulk RNA sequencing, to a smoothie. So you will use a lot of different fruits, but you will, in the end, you will see and you will taste an average. So you will not be able to identify which fruits are in there and how many of them. Whereas for single-cell sequencing, we can compare it to a fruit salad. So with this, you can identify which fruits are there, how many. You can look at the individual fruits. And in case of the sequencing, we would be able to identify, to really look at the gene expression of individual cells. So here you can see a standard single-cell workflow. So normally you will start with either with a single-cell suspension or with a tissue that you dissociate to a single-cell suspension. The suspension then has to be partitioned. So you can use it, for example, you can use wells or droplets for partitioning, and the cells are then lysed, and the mRNA is then captured and then barcoded. And then afterwards, you can prepare the library, the sequencing, and go for bioinformatic analysis. So in this type of experiments, you will be able to look at the whole transcriptome of every single cell individually. So this you can, for example, use to identify cell types or subcell types. You can discover new, even new cell types, or use it for drug treatment and drug development. So there are many, many applications possible. I will not go into any further details about the single-cell sequencing techniques and possible applications. The previous speaker in this webinar series already explained this in a very good presentation. So if you are interested and missed the previous presentation, you can watch it at any time. And but today, so my topic is our applications of single cell sequencing beyond the standard RNA profiling experiment. So the field of single cell sequencing is advancing at a very rapid pace, and there are a lot of new technologies and new possibilities to add additional layers of information to your single cell transcriptomics. So here, for example, you can see that you can, for example, look at genomics. You can then look at the correlation of genomic alterations and gene expression. You can include epigenomic information. You can, for example, look at regulatory relationships between epigenetic changes and your whole transcriptome. And these are, for example, mutations that can alter how genes are expressed, or histone modifications that can affect which parts of the DNA are accessible. And you can also look at proteomics to correlate mRNA and protein expression to be able to link genotype and phenotype. And you can also look at spatial transcriptomics to look at your cells in their positional context in the tissue. So so these are just some examples. There are many more possibilities, and so today I will focus on 3 unique products, unique kit solutions, and how they could help you in your research. So first, I will talk about our FocuSCOPE kit and how you can use this for targeted single cell sequencing. Afterwards, I will talk about our DynaSCOPE kits and how to use this to add a time dimension into your single cell sequencing research. And as a last point, I will talk about our GEXSCOPE microbial single cell RNA library kit, which can be used for yeast single cell sequencing research. So starting with targeted single cell sequencing. So why would targeted single cell sequencing be interesting? So, so in many cases for research, you have very specific questions. So you have genes that you want to look at, so you might be interested to look at the relevant gene and to really be able to get a lot of information about this gene. And so it would make sense to really be able to focus on your transcript of interest. And especially when the gene expression of this target gene is very low, then it might make sense to go for targeted single cell sequencing. And in addition, of course, targeted single cell sequencing can also give you an additional layer of complementary information. So when we go back to our fruit salad, so what would it mean in this case? So for example, when you have a fruit salad, of course, you can taste it. You can see what kind of fruits are in there, but maybe you are interested in what kind of in nutritional value of the fruits, or you're interested whether there are any contaminations or something like this. And so, and to analyze this, you would need an additional layer, and this, in case of single-cell sequencing, could be realized by the FocuSCOPE. The FocuSCOPE enables simultaneous high-throughput detection of target genes and of the whole transcriptome with high accuracy and sensitivity. So with FocuSCOPE, you can look at the whole transcriptome in addition to, for example, targeting specific mutations or targeting specific fusion genes, or look into the viral integration insertions. So basically, you can target what you are really interested in. How do we do this? So FocuSCOPE is based on our scope chip and specially designed barcoding beads. So here you can very briefly see the core of our technology, the scope chip, which is used for partitioning of cells. So you would load the cells onto the chip, and then they can be partitioned into the wells. You would load barcoding beads onto the chip, and this barcoding beads, they have probes that allow to capture the RNA of the cells. And in our case, for FocuSCOPE, we can specifically design the barcoding beads. So normally you would use poly-DT probes to capture the poly-A tail of the mRNA in your cell. But as you can see here, you can also add specific probes to really specifically capture genes. And with this FocuSCOPE, the beads can be customized with up to 10 probes. So this is how a workflow would then look like. So again, you start either with a single cell suspension or with a tissue that you dissociate into a single cell suspension. You load them onto the chip, perform the cell lysis and MR capture. And here, the important step distinction is that you will generate two libraries. So you will generate a whole transcriptome library, but in addition to that, you will also generate and enrich a target gene library. So these can then afterwards be sequenced and again analyzed via bioinformatic analysis. I brought two examples of how FocuSCOPE can be used. So the first one is the FocuSCOPE for FocuSCOPE for lung cancer. So brief introduction. So cancer in general arises from DNA mutations in the cell, which are acquired during the lifespan of a person. And many of the mutations do not play an important role in cancer, the so-called passenger mutations. But whilst the genetic mutations build up, especially in critical genes, there might be mutations that are involved in the development and progression of tumors, the so-called driver mutations. And these are, for example, mutations in genes that control cell growth or cell death or DNA repair. And those mutations might allow the cells to grow and divide at a really rapid pace and form a tumor. And those mutations are also very important to identify because some of the mutations are druggable. And so it will be really important to really know which kind of mutations you have to really be able to decide how to treat this specific case. In our case, for lung cancer, we designed Multiomics lung cancer druggable mutation panel. You can use this FocuSCOPE kit to detect lung cancer specific mutations and whole transcriptome at a single level at the same time. And so here you can see the gene panel. So with this kit, you can target the majority of important lung cancer mutations. For example, here you can see the gene TP53, which is really important and involved in regulation cell growth and cell division by monitoring DNA damage and repair. And here, for example, you can see the genes EGFR and KRAS. They are both genes that are important for signaling pathways and are involved in triggering cell proliferation. So now I'm showing some data about the focus, some experimental data for a FocuSCOPE. So the first question that we have was how specific is the FocuSCOPE. And so what we did, we designed an experiment where we mixed 4 cell lines, CCRF H1975, A549, and 3T3. And so this 4 different cell lines we mixed equally and then prepared the libraries using our FocuSCOPE kit. And so this is the UMAP that we generated. And as you can see, this 4 different cell lines really cluster well apart from each other. And and looking at this in detail, for the cell line A549, this cell line has a mutation in the KRAS gene, a G12S mutation. And when you look at the UMAP, you can see that we detect this G12S gene very, very specific in this cell line. So the other cell lines where we do not expect this specific mutation do not show this, are not, so there are no mutations detected in the other cell line clusters. This is same for H1975, which has 2 different mutations in the EGFR gene. And again, as you can see here, the 2 mutations are very specific for for this cell line and cannot be detected in the other cell lines. And for the last cell line, CCRF, where we have where we detect 3 mutations in 2 different genes. Again, you can see the mutations are detected very specific in the cell line. So we detect this mutations in the correct cells. And in the next experiment, we were wondering how sensitive this detection is. So for this, we designed an experiment where we mixed 2 cell lines in different ratios and prepared the libraries again using FocuSCOPE. Here again, you can see an example of a UMAP. And you can see that we very accurately, again, detected the mutations. And, so we can see that we can cluster this 2 cell lines apart. And here you can see that we very accurately detected the 2 mutations. So here you can see an overview of this ratios. So we tested 3 different ratios, and you can see here how much cells we captured for every cell type, and you can see that we so that this method is very sensitive. So we detected more than 90% in this in the case of the 1 to 1,000 ratio, even 100% of the cell lines that have the mutations, and which shows that this method is very sensitive. Going to our next example. This is about using FocuSCOPE in blood cancer. Normally in leukemia, so you will have genetic alterations, which leads to rapid growth of blood cells, which are abnormal. And the problem is that in this case, there will be an excessive amount of abnormal white blood cells and a lack of normal blood cells. So, so here again, there are single nucleotide variants that can be found in this type of cancer. But for blood cancer, there's also many fusion genes that are involved in leukemia. And here also for fusion genes, as well as for these mutations, some of the fusion genes are very attractive for the diagnostics and the therapy of the blood cancer. So, so here you can see our FocuSCOPE blood cancer panel. So we are targeting with this focus panel, we are targeting major SNVs and in addition 2 different gene fusion. And so this BCR ABL1 gene fusion and PML para gene fusion are both very important in leukemia. So, for example, this RARA gene is involved in regulating gene transcription, and as a PML gene acts as a tumor suppressor. And when those two genes fuse due to rearrangement, for example, of the genetic material, both the genes cannot function properly anymore. And so that's why it is very important for blood cancer. So, so next I will show you some data, some experimental data for the blood cancer panel of FocuSCOPE. Here again, we mixed two different cell lines, MB4 and U937 equally. And we prepared libraries using the FocuSCOPE blood cancer kit. And so here you can see the UMAP of cell lines cluster apart, and you can see that the two different mutations and the fusion gene are all detected, and again very specific for the cell line. And here you can see the numbers. So we detect more than 90% of the mutation on KRAS, 70% of the mutation on TP53, and 85% of the fusion genes. So next, we used the xenograft models. So we used a CDX tumor model with MB4 gene and injected it subcutaneously into immunodeficient mice to generate the tumor model. This MB4 again has this PML RARA fusion gene, and as you can see here, this time we performed this experiment with the FocuSCOPE and compared it with our standard GEXSCOPE, which uses only TD, the poly-T capturing. And so you can see here the comparison between the two t-SNE plots, and at first glance you can already see that this is you can capture far more of this fusion genes compared to when you only use normal probes to capture the poly-A tail. So here you can see that using normal poly-DT beads, we capture like maybe 1/10. We capture 1/10 of what we can capture using the specific barcoding beads for blood cancer. So to summarize, so FocuSCOPE can be really applied in many areas. So I just have some examples. For example, you can use it for drug testing and development to analyze the change in cells that contain mutation ratios before and after drug treatment. and you can use it to gain insights into disease mechanisms and tumor heterogeneity, to obtain genetic information and gene expression data simultaneously. You can, for example, use it in evolutionary biology, where you can, for example, monitor the evolutionary path of different driver mutations. So next, I will go to DynaSCOPE and how this can be used to add a time dimension to your single cell sequencing research. So again, I brought the fruit salad. So in this case, so for fruit salad, so with a normal single cell RNA sequencing experiment, you will be able to identify the fruits, but maybe you want to know how to be able to really analyze how fresh these fruits are. And so here, additional temporal resolution can really help in this question. So, for example, so RNAs are continuously transcribed and degraded, and you have so when you have a standard RNA single cell sequencing experiment, you will look at a snapshot. But this snapshot actually represents a state between new transcribed RNA and RNA degradation. So, and this RNA transcription and decay, this is very important for gene expression control, and so it's also something regulated. So to be able to really selectively regulate this, to selectively stabilize RNA, for example, or enhance and degradation. So normally when you want to analyze this, people would use multiple samplings to be able to analyze this. But with the DynaSCOPE, you can really distinguish between old and new RNA. You can really analyze transcription dynamics and regulation, and use it to research on cell type specific transcription regulation mechanisms. So how do we do this? So this is the principle of our DynaSCOPE. So what we are doing is we are adding an analogon, this S for U, to label cells. So these cells are cultivated, incubated with this compound, and then after single cell partitioning, lysis, and barcoding, what we do is we add an additional conversion step. So the S for U is converted into a cytosine analogon, and this is how then later you can you be able to have a mismatched cytosine in nascent RNA, whereas in stable transcripts you will still have the uracil. And then later, after sequencing through bioinformatic analysis, you can separate the nascent and the long-lived transcripts. So this is how a workflow would look like. You have two different options. You can use the culture version where you add labeling reagent to the cell culture, but you can also choose an injection version where you inject the reagent directly into, for example, your mouse, and then analyze this. So you will then after tissue dissociation, you will partition the cells, lyse the cells, and capture the mRNA. Afterwards, the base conversion needs to be performed so that in this in the cDNA of the newly synthesized RNA mutation sites are then introduced, and which then can be, after library generation, sequencing, and analysis, be used to differentiate between nascent and lasting transcripts. So here some data to really show that the substitution is very specific. So you can see for two cases, for lung and for bone marrow sample, that we observed this, very specifically observed this T2C mutation, which shows that the substitution is very robust. Then here you can see that we compared treated versus untreated cells. Again, this is bone marrow sample from mouse, and you can see in this UMAP clustering that the untreated and the treated cells overlap very nicely, and when you do the cell type annotation for treated and untreated samples, you can see that well, there's barely a difference between the ratios of the different clusters. So then how does this DynaSCOPE data then in the end look like? So you will be able to, so here again you see treated versus untreated with the cell type annotation, and here you see that the cells are colored depending on the RNA metabolism. So the really faint colored ones are cell clusters that are not very active, while the dark color indicates very active cells. So in this case, you can see, for example, in this sample, neutrophils and monocytes and NK cells, B cells are very active, whereas this cluster for macrophages is not very active. So, and then you can also analyze this for the different genes depending on the specific cell types. So, for example, you can see that for the first three genes, they are you find it as freshly new RNA, freshly synthesized RNA, but also as stable RNA in B cell, endothelial cells, and macrophages. While for the second group of genes, you can see that so these genes, while you can find them in endothelial cells and macrophages, those are all lasting RNAs, so there was no synthesis of new nascent RNA, but for the B cells, you can see that so you can conclude that these genes are freshly synthesized actually only in B cells. And for example, for the last three genes, you can see they are only long-lived transcripts and no fresh synthesis. So in DynaSCOPE, you can, for example, use for research into infections. For example, if you want to look into mechanisms of infection by bacteria or virus, if you, for example, want to analyze the response of different host cell types to an antigen or a treatment, you can use it for drug development and treatment. So you can really look into primary and secondary effects of the treatment on the transcription regulation. And you can also identify potential new druggable target genes. For developmental biology, you can use this to classify cell types and their dynamic gene expression and regulation processes, and use it to gain a better understanding into the roles of transcriptional regulation at different developmental stages. And of course, you can use it to look at transcriptional regulations. So so next, I will talk about the importance of single cell sequencing into the yeast research. And again, I brought fruits, but this time, as you can see, it's not a fruit salad. Because for yeast, in contrast to mammalian cells, you do not have all these different cell types. So you would, in principle, look at an isogenic population, but still, even if you look at isogenic populations, there's still differences. So, for example, stochastical variations in gene expression can lead to different behavior within a microbial population of yeast cells, and thus studying heterogeneity can provide you with important information for yeast research and engineering. Furthermore, yeast is also a very important model organism to be used for studying cellular processes, aging and disease, and it's very easy to manipulate. It grows very fast, and there's many methods and tools that you have to really influence, to change and engineer yeast. So this is a very valuable model organism. And of course, yeast is also very important in industry for food, beverages, feed, and fuel production. The challenge for yeast single cell sequencing, so the main challenge is that a cell wall is present. And so the cell wall has to be overcome to be able to access the mRNA. Yeast have different morphologies and are in general more often smaller than mammalian cells. They also have a lower amount of RNA, so a really efficient capturing of mRNA is also very important. So our GEXSCOPE microbial single-cell RNA library kit has been optimized for yeast single-cell sequencing and can be used to study heterogeneity within a microbial population. So here is just to show a comparison between the yeast kit and the standard GEXSCOPE kit. So here you can see data for a standard kit. So you can see you basically don't see any, you didn't capture any mRNA, whereas when you use the microbial kit to really break open the cell wall, you can see the difference. You can capture the mRNA. So the workflow here again is so you start from a yeast suspension. You will partition the cells, lyse the cells, and capture the mRNA on chip. Generate the prepare the library, do the sequencing and the analysis. So here's an example for an experiment. We used a mixture of different Saccharomyces cerevisiae strains, cells with different mutations, and so one group was exposed to osmotic stress, and another group was the control group. And yes, you can see again on the EU map, the two groups clustered very well apart. So here you can see the yeast that has been yeast cells that has been exposed to osmotic stress, and here the control. And so when you look at a little more in detail, you can see so here are genes that are very different between the two groups, and so most of them were genes that were involved in stress response, which also makes sense because they are usually are highly expressed in response to stress. So for example, this HSP12, this is a heat shock protein. This is involved in maintaining an organization during a stress condition, and is typically induced by heat shock, oxidative stress, and osmotic stress. Another example is the GRE2. This is also a stress responsive gene, which is usually, according to literature, induced by stress. So you can say that you can analyze this very well with the yeast kit. And here, in this case, I brought some examples of studies where people looked at yeast in single-cell sequencing. So here, for example, in this study, this group measured the transcriptomic impact of different compounds on yeast, and they were able to really find out that each compound had a distant transcriptomic footprint on the isogenic yeast cells. In this second example, the authors looked at transcript heterogeneity in single cells treated with and without stress. And they were able to really find out that even cells within isogenic populations can show significant variations in stress tolerance. So in this third example, the authors used single cell sequencing of yeast for aging research. So they did a systematic and quantitative investigation into yeast aging and studied yeast at different ages. And they were able to identify key biological processes that underlie aging in yeast. And they observed increased cell-to-cell transcriptional variability during aging. And in the last example, the authors used yeast single cell to identify regulatory relationships between transcription factors and target genes. And they were able to construct a global gene regulatory network with more than 10,000 interactions. So, so in summary, this yeast kit can be applied for many different application areas. For example, analyzing different yeast in different cell stages, cell cycles. You could use it for taxonomic research, also research into characterization of multicellular structures, for example, colonies, aggregates, environmental communities. You can use the data obtained from yeast sequencing for cellular engineering. You can research stress response of yeast populations, and you can also use yeast as a model organism to look into different diseases. So, with this, thank you for your attention. And if you have any questions.
Yeah, thank you so much for the nice talk. So we just jump into the Q&A section. Please feel free to ask your questions. Yeah, we have two questions regarding FocuSCOPE here. So can you explain more about the customized panel of FocuSCOPE kit?
Sure. So let me quickly go to this slide. So, so basically, for FocuSCOPE, we have different ready-made panels where we target very common mutations. But of course, depending on your research focus and your research interest, you might want to be able to really choose which genes you want to target. So usually then we would meet together and talk about what your goals are to together design a panel that is really specific to your needs, and then would custom produce the beads that you would need, so that you can use it for your research.
Our next question is about the capture rate for mutated sequences. Again, about the FocuSCOPE.
Capture rate. So here. So here you already get a picture that the capture is very very sensitive. So even if in the whole sample you only have 8 cells with the desired mutation, we are able to capture it, in this case even with 100%. So yeah, but the capture rate, of course, will also depend on which mutation or which gene you are actually looking at. So for example, some mutations might not be in all of your in your whole tissue. Of course, some mutations are only on one allele of so this specific capture rate would depend on your tissue type.
Next question is regarding DynaSCOPE. How long, so the duration of the treatment, effect on cell viability, and when to choose injection, when cultivation. They would like to know these points, basically.
So, okay. So when to choose injection and when cultivation. So we commonly recommend injection for studies with small animals, because then you would really look at it in their natural environment. And but if you have studies with large animals or with, for example, cell lines, then of course you will go for the cell culture version. I think the second question was about the
effect of cell on cell viability.
Okay, the effect on cell viability. So the usually we see a slight decrease in cell viability, but only between 5 to 10%. But this effect on viability, of course, also really depends on your tissue sample. So for some tissues might be affected more than others. So what we usually recommend is that during the experiment, you would really regularly check the cell viability to ensure that you don't lose too much viability.
And the duration of the treatment?
So we recommend treatment with 2 to 4 hours. It completely depends on your target. So if you know that if you already know or suspect that your gene is produced very strongly and is very active, then you would go for a shorter time, for example, even for half an hour. And if you expect it to be maybe less active, you expect the synthesis to be slower, then you will go for longer incubation.
Yeah, so I think we can wrap up the session. Thank you again for joining us. If there are any questions left, feel free to contact us and via email info@singlerenbio.com to get more informed about our product and services. Visit our website and follow us on our social media. Thank you so much. Have a great day and bye.